So thank you, Dennis, for giving me the opportunity to present my work. So my name is Assaf. I'm an assistant professor in the School of Biomedical Informatics in UT Health, at the University of Texas Health Science Center at Houston. I'm part of a new center, which is the Center for Precision Health. And this work was done with collaborators from Stanford. It began when I was a postdoc in Stanford and has continued until now when I moved to UT Health. And its cohort-specific imputation of gene expression improves the prediction of pharmacogenomic traits for African Americans. It's slightly different than the bioRxiv title because we are now under peer review and we are changing slightly some of the text. So on the agenda today, at first I will speak about warfarin dose prediction, what is the current state and what are the challenges. Then I'll explain gene imputation methods broadly using genetic variations. Then I'll describe how we use gene imputation for warfarin dose prediction. And briefly, I will describe warfarin pharmacodynamic pathway inference, which is used in this study, but for sake of flow, I will deter that to the end of the talk because it's not crucial for understanding the talk. So warfarin therapy. So warfarin, or Coumadin, as its brand name suggests, the common brand name is a common anticoagulant. It has been used for 60 years, and it is the mainstay of our anticoagulant treatment, treating more than 2 million North Americans. Warfarin works by inhibiting an enzyme called VKRC1. VKRC1, when it is inhibited, decreases the availability of vitamin K, which in turn decreases the activation of other vitamin K-dependent coagulation factors. So you can see here on this small excerpt of a graph that I took, you can see the coagulation of normal people and rate and coagulation rate on warfarin, and this is measured by measuring something called prothrombin time, which is a surrogate for the effect of warfarin therapy. The change from prothrombin to prothrombin is affected by vitamin K dependent coagulation factors, and the usual way to measure it is called INR. It is international ratio, which is a standardized time. It's not, has no units. It's just the prothrombin time over the mean prothrombin time for a normal patient. And warfarin dose must be individualized according to the patient's response. It's a tightrope between bleeding and clotting, since the warfarin has a narrow therapeutic index. So if we give too low dosage, you might risk that there'll still be clotting. If you give too high, then you run into risk of bleeding. And the protocol, the previous protocol, was to start at the middle, which is 5 milligrams per day for a patient, and then iteratively measure the INR, which is, as I said, it's the state-of-the-art way to measure coagulation rate. And every 3 days, you measure that INR and then adjust the dose accordingly. But that is, of course, still hazardous because in many cases, even if you start at 5 milligrams per day, you might run a risk of clotting or bleeding within these few days that you wait for the INR to stabilize to measure it again. So genome-wide association studies already identified very strong variants in VKRC1, which is the drug target of warfarin, and in cytochrome P2C9, which is the metabolizing enzyme, main metabolizing enzyme of warfarin. And there were such strong association that they were incorporated into an international warfarin pharmacogenetic consortium dosing algorithm. So the dosing algorithm includes several clinical components, which is age, weight, height, ethnicity, Asian, Black, or none of the two, and is the patient taking one of these enzyme inducers, the three that are mentioned here, phenytoin, carbamazepine, and rifampin. And additionally, there is the genomic part, which uses the specific polymorphisms in VQRC1 and cytochrome 2C9. So this came out in 2009 and showed the performance of the IWPC dosing algorithm. As you can see, they split the patient into three groups: low dosage, which is smaller than 21 milligrams per week, that is 3 milligrams per day, the middle one, and higher one, more than 7 milligrams per day. As you can see, they managed using the pharmacogenetic algorithm, which is in yellow, to predict for about more than 30% both of the low dosage patients and more than 30% of the high doses patients upfront the correct dosage within some range of 20% of the actual dose. As you can see, the blue one shows just the clinical features, especially for high dosage. The pharmacogenetic algorithm does much better than just the clinical. So what's the challenge? We think that there's still room for improvement. As you can see, only about a third of the patients are correctly predicted for the dosage in the beginning, upfront, and there's still room for improvement, and also identification of additional genetic factor, not just VQRC1 and cytochrome P2C9. Another thing is that the IWPC algorithm explains less variability for individuals of African descent than for European descent. And this has been known for several years. There has been recent genome-wide association study that had only minor success in finding African-American specific variants. And our decision was to test gene imputation usefulness for improving upon the IWPC algorithm with an emphasis on African-Americans. So now is the time to actually explain what is gene imputation and how does it or may solve current limitation in genome-wide association studies. So genome-wide association studies, such as the Warfarin studies, of course, are very useful for discovering genotype and phenotype associations, but as been known for several years now, they have some limitations. They often require large study cohorts to find statistically significant signal. It's even harder to detect combinations of small effect variants. It is limited, has limited transferability between population due to population-specific variants. And the mechanistic interpretation is challenging because in many cases, if you do find a specific SNP, single nuclear polymorphism, then identifying the gene that is associated with that SNP is sometimes not very clear. That SNP might be close to more than one gene, for example, or very far for any gene. So if you could associate phenotype directly with gene, you could decrease the number of tested hypotheses because obviously there's less genes than measured SNP in a typical SNP array or even whole exome or genome sequencing as today. It might bridge population-specific variations affecting the same genes. And it may also provide, of course, a mechanistic interpretation if you do understand what the gene that you associated the phenotype with actually does. For example, participate in a certain pathway. And methods like Predixcan, which I'm going to present in the next slides, infer baseline transcription levels from SNPs. And then they take these transcription levels, these inferred transcription levels, and associate them with phenotypes. So how does it work? So this is an excerpt from the paper. You use data that includes measured genetic variation and observed transcriptome levels for the same individuals. So you have n individuals on the left, and for each individual, you measure a set of SNPs, genetic variations. And also, you measure across different tissues the observed transcription levels for the same individuals. And this can be done by using the Genome Tissue Expression Project, which is a very large compendium of such type of data. Once you have that, you can build regression models to infer the transcription levels from the genetic variation. So for each gene, in each tissue, and this is T here in this small equation, you just make a regression of the SNPs, which is the X's, the alleles of the SNPs, X, and give them weights. And thus you have a model for each gene in each tissue for its expression. Once you have that, and you have a genome-wide association study, you can take the genetic variation measured in the genome-wide association study and use the regression models to transfer that into imputed transcriptome levels. So for the same genome-wide association studies, now you have imputed expression of the genes. And then you can, of course, apply any association test that you want with the trait. So that's the basic methodology of Predixcan. And we desire to use the same methodology for Warfarin and see if we can improve on the IWPC algorithm. So this is the third part. So our data included 2 independent warfarin dose genome-wide association studies with patients of European descent, and we designated the smaller number to training, and I'll explain what we actually train, and the rest for validation. And similarly, we had 2 independent genome-wide association studies with patients of African American descent. Again, we split them, define one of them as training and the other one as validation set. So we took these data sets, and for each training set and of course validation set, we first imputed the base level gene expression based on the genome-wide association studies, and of course the GTEx compendium, the Genome Tissue Expression project. And then we regressed the warfarin dose against the imputed gene expression. But now we ran into our first challenge, and that is that we have thousands of imputed genes, and currently GTEx already have more than 50 different tissues. We were not 100% sure which tissues are relevant for warfarin. We obviously knew that liver, for example, plays an important role in warfarin, but for other pharmacodynamic features, we were not sure which tissues are the relevant ones, so we decided to include all of them. So that gives you a very high dimensionality already. And we assume that many of these genes are probably irrelevant for warfarin dose. So our first step was to reduce the hypothesis space. So we first focused on 116 genes with high relevance to warfarin action. These genes come from curated and from inferred warfarin pharmacokinetic and pharmacodynamic pathways. And the asterisk here says that I will be briefly speaking about how we infer warfarin pharmacodynamic pathways, but it's not relevant for here. All you need to do is there are 116 genes that we think are highly relevant for warfarin, and we focus only on these. And also for each of these 116 genes, we took only the tissues where we found EQTLs associated with the expression of these genes, signifying that there probably is a strong genetic association with the expression of these genes. Now, obviously, and I'm going to mention that and talk about that in the summary, we might be missing some additional information, but for here, we focused on high precision, and this reduced the space to manageable 1,700 gene tissue expression pairs. So we obtained a robust gene signature from the training set using lasso regression. So we imputed the base level of the gene expression and used lasso regression to regress against the dosage, or rather we regressed against the IWPC residuals, because we wanted to show that whether we can improve upon the IWPC. So we regress not against the dosage, but against the residuals of the dosage after the IWPC algorithm prediction. And then we took that signature and tested it on the validation cohort and relative to background, which is shuffled patients, and also random signatures, where random signatures were developed from the same 116 genes, not including, of course, the genes that were in the trained signature. So we found that Predixcan signature performed very good for European descent cohorts. We obtained an R-square of 0.2 for the residual. That is, we managed to improve upon IWPC by an R-square of 0.2 with a significant p-value, but we found no robust signature for African Americans. And by robust, I mean that we ran the lasso multiple times, and we selected only the genes that remained within the lasso. We ran different cross-validation with different splits of the data, so and chose each time a signature, and only the genes that were in at least half of the runs were considered robust. We didn't find any robust gene in African Americans. So the possible explanation for that is that Predixcan uses GTEx, learns its weights, its models, regression models on GTEx, and GTEx itself is predominantly Caucasian. 84% of the population, the samples, are white. Only 14% are African Americans. So that could tell us that the bias is inherent within Predixcan when it learned the weights, the regression models. So our solution was pretty straightforward and simple, and amazingly, it managed to correct for some of these biases. We imputed the genes in a cohort-specific manner. That is, we built a model per study, per genome-wide association study, based on the SNP that were actually measured it. So for each of the 4 different data sets that we had, we learned a different model, different regression model. And as you can see here in the Pearson correlation between the means, the averages of the imputed genes, whereas Predixcan captured platform specifics, and by platform specific, I mean that if you can see on the left side, the African American cohort, which was used for training, is very different from the African American cohort that was used for validation, which in turn is more similar to European cohorts. And that is because the African American training was exome sequence, while the others were SNP arrays, genotype patient. So already that we saw that the distribution of the means was more population specific when we learned different models per cohort. So it separated well the African Americans from the Europeans. So that gave us already some hope. And now we only have to jump to the actual results. So we ran the same procedures, and here are the results of the signatures. So what you see here are the three left signatures are signatures that were validated on the European cohort. To the leftmost, you see the Predixcan signature, which, as I mentioned, managed to get an R squared of 0.2. That is the dark blue bar. And the light blue bar is the shuffled signature, the same signature but shuffled patients. And the red bar are random signature, the same size as the actual signature. To the right of that, you see two different signatures. One was trained on the European cohort, training cohort, and validated on the European validation, which managed to get an R squared of 0.08, which is about half that of Predixcan, but still significant. And I can assure you that you don't see here the p-values, but the p-values were significant, using a false discovery rate of 0.05 for all of them. And also the African American signature, trained signature, managed to be significant, even more than European trained, on the European validation. It achieved 0.1 R squared, and vice versa. The European trained signature managed to get significant performance over the African American validation set. That is the rightmost section of the figure. So the only thing that you're not seeing here is the performance of the African American trained signature on the African American validation, and that achieved about 0.05 R squared, but it was not significant relative to random signatures. We are still studying the reasons for that. We suspect it might be that we didn't capture all the relevant gene tissue pairs, and that's why we were not better than random signature, which were also taken from the 116 genes that we already know are significant for Warfarin. So these are the results. Let me show you the signatures. So what you see here to the left, you see the signature that was trained on African Americans. In the middle, it's the signature, the cohort-specific signature trained on Central European. And to the right, you see the Predixcan signature trained on Central European. So let me just point out some things here. So in yellow, you see that all signatures were able to pick up VCRC1, which again is the Warfarin drug target. And most of them, the cohort-specific ones at least, picked up the VCRC1 in liver, which is where we know it is active and where it's main point of operation. We also managed to do thyroid, and in Predixcan we did two others like heart atrial appendage. And that is reassuring. Interestingly, the SNP that is used in the IWPC algorithm was not included in the genome-wide association studies because it's already known, so they didn't measure it, and it's not included in our models. However, we do include SNPs that are in liquid disequilibrium with this SNP, thus maybe probably aiding our VCRC1 performance and appearance in these signatures. But we had many more. I think it was about 16 to 30 different SNPs that affect VCRC1 with varying strength. What is also interesting in green here is that these are genes that were already implicated in genome-wide association studies based on close proximity to the SNPs that were statistically significant. But what is interesting, for example, is that EPHX1, which is in the African American signature, was actually found in a European genome-wide association study, and GGCX, which appears in the Central European signature, was actually found in an African American genome-wide association study, thus giving us more evidence of bridging between these two populations over these genes, at least. So to summarize, Predixcan weights are biased towards Caucasians because of the population composition of GTEx samples, probably. And now we know that for sure. Cohort-specific models may correct some of this bias. Obviously, it's not a perfect solution because a correct solution will be just to measure these populations and make them overrepresented in new samples that are measured. And the third is that hypothesis-driven solutions, like, for example, focusing just on these 116 genes, may reduce hypothesis space and the number of false positives. Obviously, it might miss novel discoveries, and this requires further work. But since we had 1,700 different features, for this study, we believe that we captured the more significant expression, and we actually tested that on larger sets that did not include, did not require that there were EQTLs in the tissue of the gene. So we added all the tissues and we got inferior performance, not largely inferior, but inferior performance to that. And we got a much larger hypothesis space. And this is not mentioned, by the way, in the paper. So this is the Warfarin study, and I briefly described the Warfarin pharmacodynamic pathway inference, which is our prior work, just to explain how we do manage to pick up these 116 genes, because the Q-rated ones are much less than 116. They range about 10 or so genes. So this is called Drug Router, and that's Integrating System Biology Sources Illuminates Drug Action. It was published in Clinical Pharmacology and Therapeutics in 2014 with me and Russ Altman, my previous mentor in Stanford. And we identified that many drugs have incomplete information about the set of genes and pathway affected by the drug. And we focused on one important gene set involved in the pharmacodynamic action of the drug, what the drug does to the body, the actual action of the drug. And our objective was to build drug-specific pharmacodynamic pathways. And the motivation was that if you do find pathways of new drugs, then you can design new drugs, for example, that target new genes like STK39, which is currently not targeted by any drug. Just to explain what you're seeing here in terms of colors. So the red is the known drug target. The blue ones are pharmacogenes. These are genes that are known to be associated with differential response to the drug. So we assume that the drug somehow works through them. The yellow are the disease genes that the drug treats, and the light blue nodes are the inferred ones. So these inferred ones can be used for designing drugs or drug repositioning if you do find a new drug target. It can be used to associate genes with side effects and drug interactions. And we actually did all this in that work and suggest new pharmacogenes. And in our case, it was used to reduce the hypothesis space in just focusing on the Warfarin pathway. So I'll just go over briefly and say that we validated, we started we first explain how we build that, and then I'll explain how we validate that. So Our first observation was that protein interactions are viable for constructing signaling pathways. However, incomplete understanding of such interactions may lead to false positives. Drugs perturb essential processes in the cell. That was our observation, and many of which are already described in existing curated pathway databases like KEGG and Reactome. Indeed, we found a very significant enrichment of genes that are included in curated pharmacodynamic pathway, and they also participated in curated cellular pathways. And as input, we took drug targets, pharmacogenes, and disease genes, which are known. These are drug-specific genes which are curated and known in several databases. And we build a protein interaction network, which is constructed of two types of interactions. Those that we call freeways, which are more reliable. These include metabolic interactions from KEGG and such databases, and protein interactions that already appear in curated pathways. And we have also side roads, which are less reliable. These are typically protein interaction networks or protein DNA interaction, that is transcription factors and then their transcribed genes. And these come mostly from high throughput experiments, so we assume they are less reliable. And the way to construct the pathways is to think like optimal route computing by GPS routing applications, in which you try to proceed on freeways as much as you can, assuming, of course, the freeway is indeed free at that time and not blocked. So we start with drug targets driving to pharmacogenes. We start with drug targets driving to disease genes. And we also have intermediate driving between pharmacogenes and disease genes to other disease genes, pharmacogenes to other pharmacogenes. And the last three we regard as some kind of super shuttle, in which you might want to get from the drug target to the disease gene in the fastest way you could, but you still have to pick up some passengers, which are other pharmacogenes or other disease genes along the way. So we start with the raw subnetwork of everybody's connecting to everybody. The colors here denote the paths. So a drug target going, a red drug target going to a blue pharmacogene is colored purple. A red drug target going to a yellow disease is colored orange. And there's freeways and side roads, as you can see. And then we start with pruning this pathway, keeping only multicolored paths because we assume they are more reliable. They are traversed more than once. And then we just manage to connect them further in case they are disconnected, arriving to a final pathway that looks like that. So we constructed the pathway for 113 drugs, having at least one drug target, one pharmacogene, and one disease gene. They were robust to removal of single pharmacogenes or disease genes. As a sanity check, they clustered based on pathway overlap, which corresponds to known therapeutic families. This is a nice figure from the paper. So you can see pathway overlap. This is a matrix that shows similarity based on pathway overlap of genes. And you can see that they cluster based on the therapeutic classes like cancers and cardiovascular drugs and diabetes drugs, etc. And we also validated the inferred proteins, these light blue ones, using literature search and overlap with curated pharmacodynamic pathways. They also ranked higher in several genome-wide association studies, taking the top hits. And we also found for doxorubicin, which is a cancer drug, we did find some homology between our pathway and genes that were knocked out in yeast, give differential response to doxorubicin. So to summarize, drug router infers pharmacodynamic pathways. This pathway can be used for various applications, including drug repositioning, side effect, and drug interaction prediction. And in our current work, they were used to just reduce the hypothesis space of the question of warfarin. And with that, I'll conclude, and thank you if you have questions.